So human genome project. It is nothing but to sequence the entire DNA of the human. Okay. Take one cell, and in that cell, how many chromosomes are there? 46. Out of this 46, they will sequence 23 chromosomes. They will open the chromosome into DNA and they will find which sequence is there in the DNA, the different sequences in the DNA. That is Human Genome Project. In short, it is nothing but sequencing of the DNA of human. Similarly, there are other projects also like bacterial genome project.
So who will open the
Isolation of DNA.
inside the body only?
No, no. You have to take out the cell, isolate DNA. You all have studied how to practical is done, no? Or not yet?
Isolation of DNA.
No. Isolation of DNA is done, then DNA is used in, then they separate the DNA strands, make small pieces, and then study each piece. What is there on each DNA strand? They cut the strands into different pieces and study by piece, and then join again. That all is done using computer. Now the first point is, in this you have to know what is the aims and what are the features of Human Genome Project. The entire DNA in a haploid set of chromosome of an organism is called as genome. In human genome, DNA is packed in 23 chromosomes. Okay, this all. Now, human genome contains 3 into 10 raised to 9 base pair. 3 into 10 raised to 9 base pair. To study one base pair in this project, it was $3. Just to study one base pair, one is $3. $3 for one base pair. Totally, how many base pairs are there? 3 into 10 raised to 9. 3 into 9 zeros. 3 into 9 zeros $1 million was spent to study this. To study one base pair, $3. So 3 into 3, 9, and 3 into 3 you have to do first, huh? 3 rupees into 3.
No, I'm not doing that.
9 into 10 raised to 9. That much amount in dollars we are talking, huh?
$9 billion.
Yeah, above billion. Above billion. In dollars, that in rupees, it will be like full Goa's budget. Not also Goa's budget, full Maharashtra's budget. Full India's, you can say. That much. Now, Human Genome Project, it was for 13 years. It started in 1990 and got over by 2003. Was the first, it is called as Mega Project for the sequencing of nucleotides and mapping of all the genes. So first they will find a sequence of the nucleotide, ATGC that they will check, and then they will map where genes are present. Okay, where genes are present, because genes will give rise to protein. And any defect in human body will be changed in the gene. You know there are coding sequence and non-coding sequence. Coding sequence are called as exons and non-coding sequence are called as introns. So there is, if anything happens to introns, nothing will change. The body will be okay. But if any change is in the exon, then it will be affected. So HGP, Human Genome Project, short form, it can come, HGP. Human Genome Project was coordinated by US Department of Energy and National Institute of Health. Okay. Many countries like China, France, they had taken part in this. Don't read this one. I'll tell when to write and what to write. Okay. Okay, now you'll write. Okay, the goals of HGP, that is Human Genome Project. Now you'll write the goals of HGP Human Genome Project. First goal is to identify all the estimated genes. Okay, identify all the estimated genes in the human DNA. Okay, all the estimated genes. Around 21% something is important, where genes and where proteins are produced. 21 or 2.1%. Rest all are unwanted sequences. And these unwanted sequences, now you have got from our ancestor.
Unwanted.
Unwanted. That is introns and all, no, that we got from our ancestor like chimpanzee, gorilla. In them it is active, but in us it is of no use. That is how there is linkage. They study linkage between us and our ancestor. Whatever UTR, untranslated region and all, introns, they were active in our ancestors. Some were active in our ancestor like chimpanzee or gorilla. There. But in us it is of no use. That's why they are still there in us. That is telling us we are evolved. Second point is sequence of 3 billion chemical base pair of human genome. Okay. First to find what is the genes, but to find genes first we have to sequence. Then sequencing. Sequence mean to check AUG, ATG, that lettering, codon they will check. Next, after sequencing, you have to store this data in the database. Okay, storing of data in the database. Now this, to store this data, if you take one library, there will be 3000 books, more than 3000 books required to store this data in a library. Okay, more than 3000 books. So best idea is to store on database, that is computer. Improve tools for data analysis. Now to study this DNA, they will improve some tools. Now you all know DNA fingerprinting or DNA profiling or this checking the DNA, whether he is the son of the father of that father or not, they can find out using the
Some
DNA.
the-
Yeah, sequence, yeah, DNA fingerprinting,
For the checking for DNA with the.
father and mother. The
No,
father
when,
and son.
the child is the father.
Yeah.
Some testing for that.
Then even yeah, father and mother, that only, or even crime. They will take the DNA and they will try to find a criminal based on the DNA. Yeah. Even in small hair piece they get, they will use it. Even in sweat also they will try to find.
That if something is there of that person, then so the DNA thing they use during rape cases and stuff.
Yeah, yeah.
The ID, no?
During rape cases.
Use
Yeah.
those ones, yeah.
Transfer related technologies to other sectors. Now this technology they can take to other sectors. Yeah, crime or some other chemistry or some other sectors also. This is genome. So other molecular structure study, that structure. And last, address the ethical, legal and social issues that may arise from the project. What is ethical?
For a culture.
Somewhat like, yeah.
Social.
Like from inside you should know whether what you are doing is correct or wrong. Okay, whether it is okay. Now I cannot simply take Amaya and start doing human genome sequencing. Directly cut Amaya, take one small piece of mass and do human genome sequencing. Or I directly cannot transfer one sperm from one animal to another animal. Just for my sake, I cannot just transfer one sperm from one animal and try to transfer. The animal will might suffer. So that is called ethics. Yeah, and it should be legal also. Legally and social issues. So these were the points. I'll copy. Two approaches: express, sequence, text, and sequence of annotation. Express, sequence, text. What is tag? Tag like I'm going. We say no tag. We put a tag over there. That. So whichever sequence is expressed, they were finding with the help of a tag. Means they will make a base pair complementary and try to insert in a DNA. If that base pair is taken by the DNA, that means this base pair is present. Okay. And why it is called expressed sequence tag? What is expressed? Shown. Only which is expressed. The gene which is expressed, that only they will study. That is any gene, the exons, introns. Introns they will not study here. Because
Introns
introns of no use. That is the first step. To study what is of use, that is whatever will code for a protein. Those sequences, express sequence tags, refers to the expressed as RNA. They will be expressed as RNA. And second, sequence annotation. This is sequence annotation, which we have already studied, like introns, exons, promoter, start codon, UTR. What is UTR? Untranslated region, translated region. So just sequence the DNA and give them some annotation. You all know annotation means, what is annotation? Like giving us particular name. Yeah. Particular name we can give for a particular thing. So sequence whole set of genome containing all the coding and non-coding sequence, and later assign different region in the sequence with function. Assigning different region with function. So this first point, whatever is important, that only you find express sequence tag. Second one, completely study entire genome and write. Wrote. So write and I stop. Small means complementary DNA. So they make complementary DNA and they will try to attach this. If it is present, they will join. If not present, it will not join. And on this cDNA, they will keep some marker. Marker like X-ray, what we use, radioactive markers. So if it is joined, then it will be shown in the radioactivity. If not shown, it is washed out.
So what happens if one of the exons don't work?
Then mutation. This will not see. Okay, the procedure. Yeah, I'll explain, then you will write this. First is isolate DNA from the cell. First is take a cell, isolate DNA from the cell, then convert into random fragments. Cut the DNA into different pieces. Then clone in a host, bacteria or yeast, using vectors like BAC and YAC. BAC for bacteria, artificial chromosome. YAC is yeast, artificial chromosome. Now, why they put in this bacteria means to make clone. What is clone? Copy. So here they will make many copies. In bacteria, they will put, or in yeast, see, they take the DNA, they make the pieces. Some pieces, all the pieces they will put in bacteria to make many copies. Once you have many copies, you can easily study. Because if one is wasted, you can throw it out. You cannot keep on isolating DNA from the cell. From human cell, if you keep on isolating DNA, it will take more time. Here, what you can do, make clone and keep. Keep many copies and take out and study. Then sequence the fragment using automated DNA sequencers using Frederick-Segner method. So using computer, they will sequence the DNA. Then arrange the sequences based on overlapping region. Now here they have made cut pieces. Then here they will insert in bacteria, make many copies. Then sequence these pieces. This piece is sequence, this piece is sequence, this piece is sequence, sequence and sequence. All they are separate. Now you have to join. Now there will be something called as overlapping region. The regions which overlap each other, they can join them. Now to take simple example, how you know this part? Can you join this now? How you will join based on what? No, directly. Now if you see, I am giving you this piece and I am giving you pieces cut. Looking at the color, you can join. Looking at the first, you will see, this is orange, blue color, this is slightly blue. So this you can join. This is red, this is red. So based on color, you can join. Similarly, based on overlapping sequence, they can join the DNA. Okay. Then finally, align the sequence using computer program. Make a long stretch and align it. That is the final step. And for this, you use these two vectors, BAC and YAC. Okay. BAC and YAC. Bacterial artificial chromosome and yeast artificial chromosome. Okay. These are vectors which will make copy of the DNA. So you'll copy this procedure